Our first speaker for tonight is Professor Justin Yerbury. You may have seen him being interviewed on ABC TV's Australian Story. Justin is a molecular biologist researching motor neurone disease, a condition which he was diagnosed with in 2016. Please welcome to the stage Professor Yerbury.
Hello, I am Stephen Hawking. Last year I had the pleasure of meeting Justin Yerbury. Many of his close family members have died from motor neuron disease, the condition I was diagnosed with when I was 21. Justin has dedicated his life to studying the disease and searching for a treatment. This is Justin's story.
My name is Justin Yerbury, and I became a scientist after several of my extended family died from motor neurone disease. I had never heard of MND when it first came crashing into my life. It is quite possible that you are not familiar with it either. The most powerful way for me to illustrate what MND is to show you a video of me, just 5 years ago, in 2014, explaining how it works.
The motor system of each of us in this room controls our muscles. And it does this by sending impulses or signals from our cortex at the top of our brain all the way down to the tip of our toes and the tip of our fingers, just like this puppet. And although I'm not a great puppet master, you can see that I can move his legs and arms around. But if we cut those lines of communication between the controller and the limbs, you can see that falls limp, and we can no longer control it. If we do that to each of these, we can no longer control any of the movement of this puppet. And that's exactly what happens in motor neurone disease.
There was then, and still is now, no effective treatment. Primarily because, for a long time not enough was known about the molecular causes of the disease. I wanted to know the answer to some pretty important questions about what the underlying molecular processes were that result in MND. That is what has driven me to this point. What was very clear at the time was that protein molecules were accumulating into deposits in dead or dying motor neurons. We call the process of proteins forming deposits aggregation. So we knew early on that this protein aggregation phenomenon was a fundamental process associated with MND. I even moved to Cambridge to study with the world's leader in protein aggregation, Professor Christopher Dobson. Proteins are made as a linear chain of amino acids, and have to fold up into a particular shape to perform their function. It is like a molecular origami for our proteins. In a way, paper folding is similar to our proteins folding. A piece of paper starts flat, but can be folded up into various shapes, just as proteins start as a linear chain of amino acids, and fold into complicated three-dimensional shapes. When a protein aggregates, it loses its shape, and stacks up onto other unfolded molecules. You can think of it as being similar to stacking pieces of paper one on top of the other, into a huge tower. It is energetically favorable to stack both the paper and the proteins when they are unfolded. It would be impossible to stack paper origamis once on top of each other without unfolding them first. Even after studying this phenomenon for several years, something still troubled me. Our research told us that the process of protein aggregation has a large component of randomness. Protein aggregation relies on molecules moving around in a Brownian motion, randomly bumping into each other, to form a more ordered structure. But in contrast, neuron loss in MND is a very orderly process, seemingly starting at a focal point and spreading outwards. A clue to how to make sense of this came from outside the field of MND. I knew that there was a set of diseases that was associated with protein aggregation and that these aggregates were transmissible. These are called prion diseases. The prion paradigm stems from the infectious properties of the prion protein, which causes scraping in sheep and goats, as well as mad cow disease that decimated the British beef industry, and that crossed into humans and killed hundreds of people. The extraordinary truth was that the prion protein could change shape, and the misshapen form is capable of converting the healthy form of the protein to the pathological form. The pathological form of prion protein can spread from cell to cell, and from organism to organism, in an infectious manner, transmitting disease in the absence of any DNA, which is normally required for infectious pathogen replication. Additionally, the neuropathology of the prion diseases is highly similar to that of neurodegenerative diseases, including the hallmark characteristic of accumulation of proteins into aggregates. I thought that maybe the aggregates found in MND might also be transmissible, and that this could explain how the symptoms move in an ordered manner from neuron to neuron in MND.
Justin is trying to understand the fundamental mechanisms that contribute to the progression of motor neuron disease.
He's made some seminal observations in how motor neuron disease spreads from cell to cell, and he's come up with unique ways of understanding why motor neurons get sick.
Justin is regarded as the world's expert in the application of proteostasis to an understanding of motor neuron disease.
Proteins are an integral part of every organ in your body and make up most of the material in each cell. But it's critically important that they're recycled. So we think that in motor neuron disease, that's not happening and they're kind of just clogging up the cell.
What Justin realised is the fact that the spread of the disease, once it starts, results because these clumps of toxins migrate from one cell to another.
And so Justin is trying to understand how and why these proteins build up in motor neurons, and also how they're transferred from one motor neuron to an adjacent motor neuron, and how we might be able to intervene at that point.
10 years ago we started to perform some simple experiments that showed that protein aggregates could be transmitted between cells cultured in the laboratory. Since then, we and others have clearly demonstrated that MND proteins can behave like prions in cells grown in a dish. After publishing a number of articles on the subject, I was diagnosed with MND myself. But I needed more time, So I underwent surgery similar to Stephen Hawking, to help me breathe. It saved my life. We needed to be able to test our idea that protein aggregates could cause the propagation of the disease in the complete nervous system. While we thought about this, a team in the USA had stumbled upon a way of being able to test this idea. They tried to make a genetically modified mouse that would get MND, but it failed to get symptoms. So they thought that the mice needed a trigger to get the disease. They found that injecting a minuscule amount of brain material from another mouse with MND, or even a person with MND, would make these mice get symptoms. The disease in these mice starts at the site of injection, and travels through the nervous system. We have now sent some of our purified protein aggregates to the team in the USA, and when they inject a tiny amount of our aggregates, it is clear that the injection of just aggregated protein is enough to trigger the disease and cause the disease to spread from the site of injection. So the idea of protein aggregates being able to cause the spread of MND is true, at least in mice. Importantly, this idea explains a fundamental characteristic of MND that we can utilize to design new therapeutics. And so it is possible that a single idea and a lot of hard work could one day lead to a treatment for MND. Thank you.